Like Andy mentioned, my name is Surendra Dara. I am an extension specialist with UC Cooperative Extension in California. Unlike other states in UC or other countries, other states in the USA or other countries, University of California invests heavily in extension outreach component. That is why we have our own division. It is a division of agriculture and natural resources, and they employ people like me exclusively to conduct research and extension, because we need to educate farmers about all the new developments that we have in agriculture. So that's our focus. So I am going to talk today about entomopathogenic fungi. How many of you are familiar with entomopathogenic fungi? Okay, just two, three people. Okay, so what kind of interactions do you think entomopathogenic fungi typically have? They are pathogenic to arthropods. That is why they are entomopathogens. So the primary goal or responsibility of entomopathogenic fungi is to kill insects, ticks, mites, and similar arthropods. That's the main goal. That's how they're primarily used in agriculture or in multiple areas. But they also have other interactions. These interactions are not fully explored, and only a few of us started to study these in the past 5-6 years, and then and a lot of interest is in this area, looking at what else do they do and how they're interacting with various organisms there. So endophytic and mycorrhizal are the other two interactions. Endophyte, what is an endophyte? It colonizes these plant tissues. And what is mycorrhiza? It is a fungus that has a relationship with roots. So in addition to this entomopathogenic role, they also have endophytic and mycorrhizal interactions. And the other two are less explored, and I want to share my study results about all these three areas. So if you look at the entomopathogenic role and what do they typically do, these are fungi, not like bacteria and virus. They do not need to be ingested, and they do not act in the intestines of these insects, but they act by contact. So when you have a conidial spore, it germinates and produces a germ tube, and then when it comes in contact with a suitable host, it produces this aprosorium, that round structure. I asked for the pointer. They said they will get it, but we don't have it. But you can see that aprosorium, that bulbous structure. So they use mechanical force, and they also release some enzymes that degrade the cuticle. So they enter the insect body, invade the tissue, multiply inside the insect, and then they kill the insect host and emerge from the cadaver, and they produce more spores, and this cycle continues. So these entomopathogenic fungi are ideal for insects with piercing and sucking mouth parts, because they are more by contact rather than consuming the plant tissue by chewing. So the second role is endophytic interactions. What do they do here? What is endophyte? Typically does. Okay, it colonizes the plant tissues, and when it does, it also releases these toxins, the fungal toxins, into various plant tissues, and typically they affect the insect feeding on them, on this colonized plant, by antibiosis, antizoonosis, and similar interactions. They do not kill them through infection, but because of the toxins, their growth and development and fecundity are affected. So they indirectly affect herbivore populations feeding on the plants colonized by these fungi. So we could get some benefit and some use from these pathogens through endophytic interaction. And the third one is mycorrhizal interaction. How do entomopathogens help plants through this role? What do mycorrhizae do typically? They act like an extension of the root system, extension of the network for the root system. So they absorb moisture and nutrients better than a plant that does not have these mycorrhizae. And because they colonize the plant tissues, they can also protect the plant from some harmful pathogens. So they do have an additional role here, and this is what they could do. If you have a plant growing by itself with no mycorrhiza associated, this is let us say, I mean, this is just a cartoon showing this is how it would grow. But when you have a mycorrhiza colonizing the roots, so it acts as that extended network of roots. So it basically helps plants absorb more moisture. So that could be like a drought resistance, a drought tolerance, and also help it to absorb nutrients. So the plants grow bigger. So these are the three aspects that we have here, and I want to show one or two examples for each of those. So this is California strawberries. This is an important commercial crop in California. The industry is valued at $2.2 billion annually, just looking at the crop value. And California is the largest producer of strawberries in the world, and 90% of strawberries grown in California come from, in the US come from California. So I am responsible for one of the three regions in California, and this is a typical harvesting scene that you see. They are hand picking there, the top one, and here they're hand picking here too. But you see that big equipment they take, and that is like that is driven through the beds. So they take these berries, put them in the clamshells and put them in trays and put them on that belt. So they pick up, and it saves their time to go back and forth through the beds to give it load on the truck. Because it is a high value crop, anything, any insect or pest damaging, disease damaging could lead to significant losses. So we need to protect the crop from all those losses. And this is Lygus hesperus. We call it Lygus bug or western tarnished plant bug. It is a major pest on strawberries. What it does is it feeds on these developing berries, and because the tissue that it fed on is damaged, the berries that grow as they are growing, they develop this deformity. Deformed berry is not marketable. The quality and the taste, everything might be the same as a healthy berry, but because of the deformity, it is more of a cosmetic thing. So a lot of berries with deformity are just thrown away. And because it is in the US, they can do it, right? In other parts of the world, when I go and help them to train them to grow strawberries, even berries worse than these are just consumed normally. But in the US, everything has to look nice and clean, so they throw anything that looks like this. So Lygus bug is an important pest, and a lot of pesticides are sprayed to control it. And what happens when you overuse pesticides? Typically, they develop resistance, and not good for the environment and not good for natural enemies, and all associated risks with the overuse of pesticides is going to happen, and they frequently become resistant, so we have to keep developing new chemicals. So I have been doing a lot of research trying to use entomopathogenic fungi in mainstream agriculture, because when you look at these non-chemical alternatives, typically people think these are good only for organic agriculture, and conventional growers do not see the importance of these alternatives, and they do not want to use them, thinking that they're not as effective as chemicals. So I have been trying to develop IPM strategies where you rotate and combine these alternatives with chemicals so that you have a true IPM program. So here I am going to show the treatment list from my last year's study. These are 12 treatments. All the ones in black are chemical pesticides. I know you may not recognize those names because these are trade names, but the numbers next to them indicate the mode of action group. And the idea of this treatment, you see each column is one application. Like these were applied at weekly intervals, and then I am trying to rotate them, different modes of action groups, and also combine. The blue is one fungus, entomopathogenic fungus, Botanigard is Beauveria bassiana, and the green, Met52, is Metarhizium bruneum. This is another fungus. and then the brown one here is diatomaceous earth, and then the orange one, Moltex, is azadirachtin. So azadirachtin acts as an insecticide, repellent, and insect growth regulator. And then Rimon here, see that number 3 and 4, you see Rimon, that is a novalduran. It is also a chemical growth regulator. So using these things, these botanical and microbial materials as an alternative to chemicals, and developing a kind of a combination. So as you can see here, there is no end to these combinations that we can think of. It could be another 200, 500 combinations, but we can only do so much. And this is a study done in commercial strawberry field, so we have some restrictions and limitations on how much you go, and it is very expensive to do these studies too, finding technicians and finding room and space, everything. So these were applied. This is my experimental plot. These are 4 replications. You can see 4 blocks, and each treatment had 75 feet long beds that are 7 of those within each plot, and then we took the counts in the middle 3 beds. So it's each treatment is about a quarter of an acre, and that's a very large area. So we applied, and we took the counts every 5 or 6 days after application, and these are my technicians looking at these insects and whatever arthropods they could collect, natural enemies and pests. The top one we try to apply in the evening, because when you are applying fungi as pesticides, the whole night of a cool and dark period, like 12 or 13 hours or more, that helps the fungus to establish in the system. And this is a diatomaceous earth powder dust formulation applied by a backpack duster. And if you look at this, this is the summary of all Lygus life stages, nymphs, older, younger nymphs, and adults from 3 applications. Because this is a commercial field, you cannot have uniform populations. As you can see, there is a significant difference in populations before the treatment started, and as it frequently happens, after you start the treatments, all those differences disappear. It is a big challenge we always have in field studies, but that's what we have. But when you look at this and see that, oh, okay, there is no difference in treatments, that's not true, because the initial population is not uniform to measure what actually impacted. So if you look at the difference, percent change pre-treatment and post-treatment, this is what we have. So you can see some differences there, and wherever I have these blue and orange and green dots, that indicate that is where non-chemical alternatives were used. So the sequoia high, see the one on the far right, that is a new chemical from Dow Agrochemicals, and it is a very effective material against Lygus bugs, and obviously it worked fairly well. None of these materials reduce the populations below 0. They were only trying to prevent the buildup, but that is still fine because farmers need some kind of solution, and this is what we have. So if you look at the numbers, actual numbers, the chemical that is like a standard that provided control with limiting the population buildup to only 14%, and right next to it you have these non-chemical alternatives. That one is with 17%, very close to it. So that means, even if you go to number 4, that is also fairly close. The idea here is to show non-chemical alternatives can play an important role, not just chemicals, in reducing pest populations. If you look at number 7, that Rimon and SAL, Rimon and Sequoia and Rimon, they're actually very popular combinations and popular, effective, supposed to be effective chemicals. But if you see, populations increased by 167%. So again, the highlight of this is that entomopathogenic fungi, are important in pest management. As we can see here, that Botanigard, one fungus, Beauveria bassiana, and Met52, another fungus, both of them, when used in this kind of rotation, they work fairly well. And then coming to the next part, endophytic colonization, I conducted this study in 2010 where we were inoculating these potted strawberry plants with these two fungi and see if the fungus colonizes the strawberries. So we did not have any literature showing that these fungi colonize strawberry plants, so this was the first study. So we inoculated the potting medium and collected samples periodically, looking at basically the tissue. So we took the leaf material, plant material, roots and petioles and everything, and leaf lamina, and surface sterilized in bleach, and then cut the tissue and plated on selective medium, and then after the final rinse it, we also plated on these selective medium to show that the sterilization process worked, and it actually sterilized everything on the surface, and whatever fungal growth we see from the tissue is because from inside because of the colonization. So this is what I had. Fungus Beauveria bassiana emerged from roots, petioles, leaves, all leaf tissues, and it persisted actually for 9 weeks after we inoculated. So that's a very long period. So this is from controls that fungus did not grow, and whatever you see the growth, they are like saprophytes. So it is not the entomopathogenic fungus, to show that we did not have any contamination there. So now that this experiment proved that this fungus colonizes strawberry plants, what is the goal? Our goal is to see if this fungus has any impact on insects feeding on the strawberry plant. So we conducted another study where this is also a greenhouse study where we had untreated, and then fungus inoculating soil only, then foliar application of Beauveria bassiana, and then foliar and soil application. So this is what I have in terms of live green peach aphids. We used green peach aphids as a model to see if it has the fungus inside the plant has any impact. You do not see a lot of difference, statistically significant difference between untreated and soil treatment, but you can see that when it was applied to the soil, the fungus colonized, as we saw in the previous experiment, but it did have some impact on the survival. And then if you look at the soil and foliar, because it is direct contact, it kills more. I will also show the dead ones here, so it is easy to understand. So foliar, because it is direct contact, so you have insects dying there from this treatment. Then soil and foliar. Technically, if soil is causing this much and foliar is causing this much mortality, when you have both of them, shouldn't you have a bigger control? Technically, right? It is like an additive effect. Then why do we have more surviving in the soil and foliar treatment? That is because this fungus is actually promoting plant growth. So when we apply to the soil, it is promoting plant growth and supporting more aphids there. So although it is killing more or controlling more, at the same time it is actually has a positive effect there. So this is why that this is kind of shows what is happening there in the next experiment I am going to show. So this is another experiment where I used Metarrhizum bruneum. Here, same thing applied to the soil, and this experiment was actually not intended to see if it has any impact on mites. I had accidental mite infestation in the field, so that is in the greenhouse, and it was actually interfering with my study. But what I noticed was those untreated plants suffered more from the mite damage compared to the treated plants. You can see how healthy the treated plants, although there were mites. And you can see it is like 10 and 14 days after the damage, there were, I do not have any statistical data because this was not a study intended to study that impact. But clearly you can see when the fungus was applied to the soil, it probably helped either through antibiosis or by helping plants, absorb more water, retain water, and through that kind of activity. And then finally, what kind of mycorrhizal interactions do we have? Basically mycorrhizal interactions help plants to protect from pests and pathogens, and also absorb water and nutrient and grow better. And this is a study I conducted in a Los Angeles County prison. 2 inmates actually worked on this project. It is a special prison reform program. They participated in the program. And we had those 3 treatments, untreated, and the second one is microbial growth enhancer, which is supposed to promote the plant growth, and then I had Beauveria bassiana. And they did not know what material they were receiving, so they just applied and took the plant measurements, health counts for the next few weeks, and they rated based on this, dead, weak, and very good based on the health status. And this is what we had, untreated. And then that HYTA is a commercial product meant for this purpose, and then Beauveria bassiana. Surprisingly, plants treated with Beauveria bassiana performed better than the other treatment. So this led to a field study. Because my time is coming up, I quickly want to show this, where we had a commercial standard with no Beauveria bassiana application, and then Beauveria bassiana applied periodically, 7 weekly applications and 6 biweekly applications at the base of the plant. And this is how they were growing, and we took the canopy measurement and yield for the next few weeks. And if you look at this, initially Beauveria bassiana treated plants, there were no statistical differences there, but Beauveria bassiana treated plants grew slightly bigger the first 2 times. And when we looked at the yield, it did not really help initially. It was a kind of that the fungus treatment is lagging behind, but towards the end there was a slight increase in fungal treatments. So in the yield, and this is for total strawberry yield, and this is for marketable berry yield. So I repeated this with more treatments and more strategies. So we just concluded that study. I did not include that here, but conclusions of these 3, basically to show that entomopathogenic fungi play a bigger role, not just to kill insects and mites, but they play a bigger role in promoting plant growth and health. And if we understand these interactions better, we can make a better use of them in crop production, and they contribute to a better pest and disease management and crop production through sustainable management. With this, I conclude my talk, and this is an IPM Info app that I developed for iPhones. If anybody is interested, it is free to download, IPM Info. And thank you. I don't think I have any time for questions. Thank you for your opportunity. Okay. Yes.
Yeah, I missed your biodata. Please let me know exactly you are an entomologist.
Yeah, I am an entomologist.
Graduated from where?
It is in the book, and you can see that. Yeah.
All the chemicals which you have used over there, they are available in the market, and the farmers can afford them. What is the prices of that?
They're all commonly used pesticides there. Like I don't know the prices of everyone, but they're all in labeled, registered products. They frequently use, but like I was saying, they prefer to use chemicals only. They don't use these non-chemical alternatives. So that is one of the reasons to do these studies and show, okay, you can use them also, and they're equally effective. Yes. Yes, sir.
In which tissues actually are they living as endophytes? As in which plant tissues, in the leaves or in the roots?
They were in roots, and they were in petioles, pedicels, and everywhere.
Yeah, when you say roots, can you say it more precisely as where in the roots?
Where? No, I did not check that, because that was already a laborious and a big project. So I was trying to see, do they grow or not? And then I know, is it in intercellular or intra and all those things? We did not study that. Yeah.
Last question.
Yes.
This fungi colonize very well the strawberry plant.
Yes.
And you identify between what strain you apply,
These are commercial strains.
but what is the name of the strain?
GHA and F52. And one of the reasons I do studies only with commercially available materials, because farmers can use. If I use any private isolates, Yeah, they do show that these work, but farmers cannot use them unless they are registered products. So all of the materials I used are commercially available, registered for strawberries.
Okay,
Yeah.
we also work on it in the strawberry plant.
Uh-huh.
It was very, very excellent results, and the colonization is high, more than 80%.
Very good, very good.
Then when we apply, it's very important for your project to you determine the total percentage of colonization before your project apply, and after you evaluate how is the availability of the
Yeah.
colonization of plant.
It was there, but I did not show. It was already published.
Okay.
Because this is only a 20-minute talk. I only showed the highlights, but we have how long it colonized and what percent it is. It is already published and.
Before the project and after the project.
Yeah.
See how is the growth
Yes.
availability and how percentage is lost.
That's right. Yeah. Yeah. Very
Congratulations,
good.
you
Good
were
point.
excellent
Yeah.
presentation.
Thank you. Thank you all.